Okay, so we'll continue our discussion on the in vivo biocompatibility, that is the preclinical studies on biomaterials. Um, we we have discussed in the last module that what are the biological events that take place at the implant tissue interface. Particularly this slide I have spent significant amount of time to make you understand that this interaction first starts with this attraction of neutrophils towards this implant, synthetic implant, followed by similar attraction for fibrinogen, protein adsorption. This acts as a precursor for the adhesion and functionality of fibroblasts on the protein adsorbed layer. Simultaneously, the other cells like monocytes also adhere along with this fibroblast. Now, once this large number of fibroblasts they are functional on the biomaterial substrate interface, then they will secrete collagen, and then it will form a collagenous matrix of the extracellular collagen based extracellular matrix, which will ultimately shield the biomaterial through the formation of the fibrous encapsulation. And along this time scale, just before the fibrous encapsulation is matured, then foreign body giant cells also activity also become prominent, as well as that macrophages. So, this entire thing takes place over a period of 3 to 4 weeks. So, this is the time of post implantation. Now, what I will do in the first half of this module, I will just try to finish the scientific discussion, and with certain examples taken from the literature, just to make you understand that how this in vivo response can be characterized in the context of the biomaterials, in the context of orthopedic biomaterials particularly. So, essentially this fibrosis and encapsulation involves two separate processes. One is the replacement of tissue by parenchymal cells at the same time, and second one is the replacement by connective tissue that constitute the fibrous capsule, as we have seen before. And these processes essentially are controlled by the cell growth capacity in the tissue receiving the implant and the persistence of the tissue framework and degree of injury. So, these things has been mentioned very clearly in the last slide also. Now, other things you can see here, this is that histology slides, and this part of the slide is essentially hematoxylin and eosin stained region, where you can see lot of cellular activity at the interface of the biomaterials and the tissue. Now, two ways you can evaluate the tissue response. One is the in vivo degradation, that can be quantified using volume changes or changes in volume of the implanted material as a function of time. The second one is the nature and distribution of vasculature, and here I may like to recall you the two concepts that I have mentioned in the last module. One is angiogenesis, and then second one is that vascularization. These two concepts were defined in the last module. They appear to be synonymous, and this is particularly more relevant, this both the concept for the porous tissue scaffolds. Essentially, they are quantified by new blood vessels that are formed into the porosity of these 3-dimensional interconnected porous scaffolds. And so, how these blood vessels are growing inside, and so that this blood and oxygen can be transported to the cells, as you can, if you recall this kind of picture that I have shown. And at the same time, that how these blood vessels are branched out, so that different these blood vessels, they can penetrate more and more into the different different tortuous porous path. So, these two things can be quantified by this angiogenesis and vascularization, and many times in the tissue engineering people do use angiogenic growth factor just to stimulate this process of angiogenesis. So, before implantation people use angiogenic growth factor so that this angiogenesis can be promoted once this tissue engineering scaffold is implanted into the animal tissue. So, that is what I have mentioned, that this is that nature and distribution of vasculature also is important, and this also has a relevance for sustenance and metabolic function of the infiltrating tissue. Now, the three more factors which are mentioned here. So, altogether there is multiple factors which influence this angiogenesis, multiple factors which influence the tissue response. Now, among these two factors were mentioned in the last slide, and this slide certainly mentions or summarizes three more factors. Third one is the nature and distribution of inflammatory cells, including neutrophils, monocytes, macrophages and fibroblasts. Now, this kind of this requires lot of quantification based on extensive analysis of a number of histology slides. So, essentially you can see that how these different kind of cells which can be clearly marked based on the appropriate staining or based on the appropriate tissue or cell specific tissue specific staining agents, and their quantification in terms of this, how this number of cells in terms of the presence or absence and the time frame of their occurrence, like at what kind of time frame time scale after the post implantation that fibroblasts keep on increasing or they decrease, and how this changes takes place over time frame, that indicates the nature of. So, all the cellular activities essentially constitute the inflammatory reaction. It is important to recall that irrespective of biomaterial composition, irrespective of the way a biomaterial is placed in any given experimental animal through surgery or by injection of this biomaterial eluates, inflammation cannot be avoided. Only the degree or the extent of inflammation can be modulated or can be reduced through certain through particular to the use of particular biomaterials. So, therefore, inflammatory cells or presence or absence of different other cell types, as we mentioned neutrophils, monocytes etc., their quantification is indeed necessary, and this quantification is more important so that one can develop both a qualitative and quantitative understanding of the tissue response upon implantation of an implantable biomaterial. Okay. I hope I have made this point very clear. The fourth point is the thickness and quality of the fibrous tissue. Now, this thickness and quality of the fibrous tissue means that this actually indicates the degree of the acceptance of the implanted material in the osseous system. So, essentially this thickness certainly would increase with time. This thickness of this fibrous tissue certainly many times goes linear or many times goes in a non-linear manner with the time in implantation. Now, other parameters which one can also look for while analyzing the histology slides is that the signature of calcification, necrosis and apoptosis. So, remember necrosis and apoptosis is the two ways that cell death can take place, and which have been introduced or discussed quite a significant extent in some of the preceding modules. Now, these are the two examples where a biomaterial where you can see this biomaterial has a little bit irregular shape. It was implanted in an animal, and according to ISO 10993-6 implant effects, you can see there are certain regions of the inflammation. So, you can see that region of inflammation. So, this is the region of inflammation, and this is the region of inflammation, and certainly there the tissue structure or the appearance or morphology of tissue is certainly different from from a area which is a remote from the implant. Now, side by side you can see that another another material of similar shape or size, but of different composition, when they are implanted initially in this particular shape, you hardly see any material left after the implantation for same material for same time period. For example, if this implanted for 12 weeks in the same material, the other is implanted in this 12 weeks for the different material. So, let us say this is a material A, this is a material B. So, you would like to see that which kind of material exhibits minimal response or severe response. Remember, minimal response cannot be avoided. So, whatever is the material composition, I repeat, there would be inflammatory response. Now, when you compare this kind of situations, you have to make sure that you are using same animal strain. Like if you use that New Zealand rabbit, New Zealand male rabbit, so you have to use both the cases New Zealand male rabbit. Second important thing is that you have to also use similar kind of size and shape of the implant material in both the cases. Only thing you are allowed to change is that chemistry or chemical composition of A and chemical composition of B. So, therefore, you can compare now what is the tissue response in case of A and what is the tissue response in case of B, and certainly you can see in case of B the material is almost gone. So, it leaves only certain traces of the material B, and also if you see that larger area, much larger area which I am tracing by dotted arrow, this larger area of the tissue is now inflamed or is showing the signature of severe inflammatory response. So, certainly material B shows more biodegradable in nature. So, it certainly shows reflection of biodegradability as well as severe response, whereas this is more like biostable or bio inert kind of behavior. So, from this tissue response also, on the basis of tissue response, you can essentially distinguish between two type of response. In one case you see bioactive, biostable or bioinert response, another sense is kind of biodegradable response. In one case left hand side material shows minimal response, whereas right hand side material severe response in the same animal strain. Okay. These are some other examples which is shown for hydroxyapatite coating. Remember, this hydroxyapatite coating is widely used for on the major orthopedic implant alloys like titanium, 6 aluminum, 4 vanadium or 316L stainless steel stem. So, this hydroxyapatite coating, depending on the way these coatings are deposited on the substrates, whether it is called electrolytic deposition or whether it is called plasma spraying. Plasma spraying, by the way, is commercially most widely used. Electrochemical deposition, as the name suggests, that it takes place more in the room temperature deposition conditions, whereas plasma spraying involves very high temperature deposition of hydroxyapatite on this titanium 6%, aluminum 4%, vanadium or stainless steel substrate. In both the cases, they are implanted for 14 days in rabbit animal strain, and this was reported in the journal Biomaterials almost 10 years ago. Now, what you see here, that if you consider that the left one PSHA stands for plasma spread hydroxyapatite coating, there is very clearly you can see that the evidence of this bone tissue material tissue interface, and also you see clear features of the mineralized tissue. Mineralized tissue means it has calcium phosphate content. Whereas in case of electrolytically deposited hydroxyapatite coating, this similar evidence is also observed, but here the interface is more diffuse interface. Here again that mineralized tissue forms, but the interface is clearly more diffuse interface. And this is that these images were taken in the transmission electron microscope and by bright field imaging, and prior to the transmission electron microscope these tissue sections were stained with a particular staining agent like uranyl acetate and lead citrate, so that easier visualization is now possible, and so that one can confirm the mineralized tissue formation. Some more examples, nickel titanium, this is the classic case for the nickel titanium. Now, what is the relevance of the nickel titanium? Nickel titanium is used for the cardiovascular applications as stent of cardiovascular applications, and people use certain coatings nowadays like ceramic coatings, so that cardiovascular applications that by hemocompatibility or blood compatibility is very important, as I mentioned earlier in one of the earlier modules. And to improve the blood compatibility, one has to use certain ceramic coatings, and some of the coatings that I have mentioned earlier, if you remember, one is the diamond like carbon coating or titanium nitride coating. Now, this diamond like carbon coating or titanium nitride coating are normally deposited on the titanium or nickel titanium substrates to improve their hemocompatibility property. So, next level question therefore arises whether this kind of coating deposition on nickel titanium will have some influence on the way tissue formation takes place, or way tissue compatibility is influenced in the in vivo conditions. And for that, again these implants were put it into rabbit animals for 13 weeks. Now, on the left hand side you can see certain bulge of these cells here like bone cells, and collagen on the this is the uncoated one, and this is the right hand side is that coated nickel titanium. This is the coated nickel titanium, and what you see on the right hand side, that you see that more number of bone cells. This is the osteoblast cells here, and also this is the collagen fibrous network. This fibrous network actually essentially signature of that extracellular matrix deposition along with the bone cells on this particular substrate, that is ceramic coated nickel titanium. So, in both the cases I have mentioned that if you put coatings, certainly this tissue response is enhanced. Okay. Now, having given this background on the tissue response in terms of that scientific understanding, I will now explain some of the important issues related to how to design this kind of preclinical study in terms of how to select animal models, which kind of animal models are to be selected for what kind of experiments, and second thing is that what kind of defect model is to be used for particular implantation. This is the typical list of the animal models which are widely used in biomaterials research. Now, typically, so the first two mice and rats, these are like small animals, and why rabbits are written in different font color, just to highlight the importance of rabbits, or just to highlight the fact that the rabbits are by far most widely used in bone tissue engineering research. since the name of the course is Biomaterials for Bone Tissue Engineering Applications, therefore, I wanted to highlight that which particular animal strain which is used most extensively in bone tissue engineering applications. The other relatively larger animal models is the dogs, goats, sheep, pigs, and out of that sheep and pig model is the larger size animals, and they are more used for cardiovascular research, particularly for the heart valve applications or any kind of design related issues in the heart valve. When one needs preclinical study, then one has to use the sheep and pig model. Other things I will also mention later, or perhaps while summarizing this preclinical study, is that even if some materials are tested for their preclinical efficacy in the small animal model, then ideally that material's biocompatibility is to be subsequently proven at still larger animal, or in other words, simply carrying out experiments or conducting preclinical experiments in small animals is not sufficient enough to claim the in vivo biocompatibility of that particular animal, or on the basis of small animal model experiments only clinical trials in human patients should not be conducted. So, therefore, therefore researchers are encouraged to conduct these experiments to start with small animal models, then validate the small animal model biocompatibility results in a relatively larger animal or medium sized animal, further to further plan for the clinical trials. Okay. So, animal welfare requirements is one of the key things. Basically, now I will show you that how to kind of what are the perspectives on which animal preclinical study needs to be designed, and what are the factors need to be used or need to be considered while designing these kind of animal experiments. So, in terms of animal welfare requirements, I mean which essentially vary from country to country. First of all, any animal testing facility must be approved by a nationally recognized organization, and therefore, one should have institutional ethical committee in place. Institutional ethical committee also should be approved by committee for the purpose of control and supervision of experiments on animals, which is abbreviated as CPCSEA. Now, this animal ethical committee not only consists of veterinary surgeon who actually does these animal experiments, but also consists of biologists, any lay person or common man from the society, as well as a lawyer who can who knows the rules related to the animal ethical committee guidelines and so on. Now, in accordance with the appropriate regulations dealing with the laboratory animal welfare, one has to subsequently follow the different guidelines like ISO standards, like ISO 109932, that is biological evaluation of medical devices, that is part 2 of the animal welfare requirements. Okay. Now, ideally all the animal testing should be conducted under good laboratory practice, GLP, quality per platform, as per US Federation of US FDA rules. That GLP compliance is necessary. So, I need to highlight this point, that GLP compliance necessary for animal experiments. Okay. Now, these guidance documents, there are several guidance documents, and various countries they have their own. For example, ASTM, American Society for Testing and Materials, ANSI, that is American National Standards Institution, you have British Standards, you have ADA, American Dental Association, and we have also have BIS, that Bureau of Indian Standards. Again, I have emphasized the first one, that is ISO, which stands for International Organization for Standardization, and ISO testing requirements is by far the most widely used in animal preclinical studies. Now, there are different depending on what kind of biomedical applications you are targeting. For example, if you are interested for cardiovascular applications or cardiovascular implants and artificial organs, then you have to follow ISO or DIS 8637 guidelines. If you are interested for dentistry, orthodontic or prosthodontic kind of applications, then you can use the guidelines ISO 7405. Apart from that, published literature which are kind of available in peer reviewed journals. So, those kind of literature also can be used to study to design the animal experiments, and recommendations of the scientific committee like Institutional Animal Ethical Committee ISEC. They can propose certain guidelines to modify or to add to the proposal that an investigator has submitted for approval. So, those recommendations also need to be taken care of before conducting these animal experiments. So, one of the things people do to at the very beginning is to just to see that to assess the biological safety of any medical devices or large animals or large material devices, and for that it is recommended to use large animal models, and this is the chronic device implantation in the tissue of final application with human sized devices. Now, why I am kind of emphasizing with human sized device? Now, normally in the laboratory scale, always we use some scale down samples or samples of simple geometry or smaller in sizes. Now, human device sizes, for example, if you are interested on acetabular socket, then human device size you have to use the same device size as well as femoral ball head of same size. Okay? And therefore, it is important to use same device size, so that acceptance of that human sized device can be better assessed in a relatively large animal models. Since you are using human sized device, one cannot use small animal models in this particular case. Now, post fabrication procedures before this material can be put it into the human patients, that involves sterilization, packaging and storage, and sterilization can be done in different way like UV sterilization and so on. So, I think I will stop now and then I will start with this next one in the next module.